So I think we'll begin. My name is Andrew Pepper, and it's an absolute pleasure to moderate today's ATI seminar. We'd like to start off by the university acknowledging that we are located on Treaty 6 territory and respects the histories, language, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. We would also like to thank Paladin for their support of the ATI seminar series. Well, I'm very pleased to announce and introduce today's ATI seminar speaker, Dr. Jayachandran. Dr. Kizhakkedathu is a professor in the Department of Pathology and Laboratory Medicine and a member of the Center for Blood Research at the University of British Columbia. His research encompasses multiple themes, including cell surface engineering, macromolecule therapeutics, and blood-compatible surfaces and devices with a focus on biomaterial innovation with a common foundation in applied hematology and translational medicine. Dr. Kizhakkedathu has received numerous awards and recognitions, including the recent nomination as a Canadian Research Chair Tier 1 in 2021, and named a Fellow Biomaterial Sciences and Engineering in 2020, and the UBC Killam Research Prize in 2017. He is also honored with the CIHR's New Investigator Award in 2005, and Dr. Kizhakkedathu's research has been published in numerous scientific journals, over 200, including some pillar journals, Science Translational Medicine, Nature Materials, Nature Biotechnology, Nature Biomaterials and Engineering, Nature Communications, Blood, American Journal of Chemical Society, and Biomaterials. So welcome, and the floor is yours.
Okay, very good. Okay, good afternoon, everyone. Actually, it's a pleasure to talk today, and thank you, Dr. Pepper, for the kind introduction. And today, I'll be talking about one of my recent work on organ engineering to prevent transplant rejection. As Dr. Pepper already mentioned, I'm from the University of British Columbia. Before beginning, I'd like to disclose a few things, and some of these companies we founded from our own work. But today, I'm not talking about any of those in this talk. So my lab, before is to introduce my lab. My lab is a biomaterial lab. It's looking at developing technologies and treatment for different unmet clinical needs. And we are developing novel materials and molecules in relation to blood. And we have 4 pillars of our research, including cell surface engineering, therapeutics, and blood compatible surfaces and proteomic reagents. And I'll be talking about one aspect of this project, like cell surface engineering today. So this project started on the GLYCOCALYX editing or engineering. Started with a graduate student in 2014, and recently, actually, we have the first of those things came out in Nature Biomedical Engineering. And currently, actually, like this is continued by a graduate student, Daniel, and then another postdoc, and 2 other graduate students actually recently started on this work. So let's start with the immune system. Right? And so and I don't have to give a detailed understanding about the immune system here, actually, as you are already working on some aspect of that. And so the immune system consists of different cellular players, which is in the circulating blood, as well as protein players, as well as the endothelial cells, actually. So it's act as a kind of like defending us from the pathogen invasion, and also kind of clearance of damaged cells or tissues in the body. So you also have kind of like there's another clear role of vascular lumen or endothelial cells or vascular the vasculature, and the endothelial cells on the vasculature is also play an important immune regulator. It can communicate with immune cells, as well as it can communicate with the protein players in the immune system, so that actually like you have normal, healthy, balanced immune response is going on. And this endothelial cell lining consists of kind of like there's a lot of different types of proteins, as well as proteoglycans and glycoproteins are expressed, which are also kind of like which provide these endothelial or cells provide an important function as kind of an immune regulator. So when things goes wrong, actually either endothelial cells are getting activated, or kind of like it's a damage happens, and you can get recognition of kind of like immune cells around the endothelial cells, and then it could adhere to the endothelial cells, and then transmigration. And this could happen if there's a damage to tissues, or there's infection in the tissue, or any kind of like other complex things happening. And this immune recognition can happen due to there are direct recognition of like the players, like actually antigens, or to the right players, or kind of like other kind of like indirect recognition, or kind of like antibody mediator recognition, including kind of like opsonization of the transplanted cells, or opsonization of the damaged endothelial cells, or kind of like and this has kind of like a connection between our adaptive immune system and innate immune system. And all these players are kind of important in terms of recognizing the foreign materials or foreign tissues to be transplanted, or kind of like, it keeps the cells happy, or tissues happy within the system. And currently, actually, there are multiple ways actually can kind of like challenge this, or kind of like make this immune system. It can suppress the immune system, like to be for a beneficial effect, and including immunosuppressants, where actually like mostly these immunosuppressants targeting on T cells or other immune players. And also one could utilize kind of like gene editing actually here. Especially like T cell editing or B cell editing, and to beneficial aspect of that actually, like so that the transplant is kind of like survive more. Another things actually one could potentially do, people have done actually, like cage the transplanted cells in a kind of a biomaterial, so that there is no kind of like interactions between the protein players or the immune cells to this transplanted organ, so that it could potentially develop a synthetic organ in mostly kind of like in islet transplantation. This has been used. And another today actually, like another approach actually, like looking at immunoclocking actually, whether it could create a barrier, actually a thin barrier, so that actually like this barrier can interact with the immune system in a way that it could provide the beneficial effect of that. Today I'll be talking about kind of one of the aspects of this immunoclocking approaches using cell surface engineering. So cell surface engineering actually like is a kind of I could say it's an engineering method or a chemical method to modify the cell surface or the cell itself, so that actually it can generate a kind of a rapid and selective attachment of biomolecules on the native surface of the cells. And since the modification or the cell surface is really complex, you need to have kind of like a really very selective or selective modifications, so that the biocompatibility or the surface is is not damaged, or kind of like it keeps the native structures. So one such approach, like people started in the early stage, islet transplantation, like looking at modification islets, and we have used kind of like similar kind of approaches for kind of attaching polymers on the red blood cell surface, actually, which started many years back, and shows that we might be able to kind of protect the antigens on the cell surface, and you can also kind of like do kind of like provide kind of a coating, like such as a hydrogel coating on the surface, so that it could possibly work. And also one could use kind of an enzymatic strategy, very specific strategies, so that it could, if it is a sugar-based antigens or carbohydrate-based antigens, one could possibly cleave. And such a recent approach actually we developed in our lab in collaboration with Dr. Videsh on kind of like cleaving A antigens and on the red blood cells to create universal red blood cells. So this kind of approaches, kind of like whether it is a very specific approaches or enzymatic approach, will provide a platform for the modification of cell surface so that it can interact with the immune system in a positive way. So what are the technology applications of this kind of things? Actually, one could potentially use it in transfusion, and it could also potentially use a therapy for the modification. If you could do like endothelial cells modification, so it could use as a therapy, and also it could utilize it for transplantation. So that is what actually like today I will introduce it to. So why in organ transplantation? So you have, as if you in the case of transplantation, a donor is kind of donating a organ to the transplanted to the recipient, and during this process, actually, there is a lot of things can go wrong. One is kind of like majorly actually the blood vessel is shown here, and it could in the normal case actually the blood vessel is intact, and this the vasculature or the vascular surface is kind of like really intact there. But during the process, actually, there is things can go wrong. To mitigate that or mitigate that, you possibly use immunosuppression, global immunosuppressions, which could have like potential side effects. And also kind of like this injury happening during this transplantation or early preservation, and it could lead to delayed graft function or antibody-mediated rejection. There is kind of a clear evidences or there are clear connections showing that kind of like ischemia, the early injury is kind of like leading to kind of many of these bad effects. And looking at the vasculature, it little more closely and endothelial cell vasculature. So you can look at in the normal healthy endothelial cells or the blood vessel, you do have a lot of different molecules, as I mentioned earlier, which is expressed, including heparin sulfate, carbohydrate sugar kind of like ligands, sialic acid ligands, or other kinds of like ligands expressed on these endothelial cells, actually, which prevent or it can act as kind of a immune regulating organ there. But in the case of like during the transplantation or during the preservation process, like this the healthy endothelium actually like it acts as kind of like a good barrier, and it can act as kind of like not only kind of a steric barrier, also it can act as a mechanical protection, and also kind of like it could like it possibly kind of like generate kind of like immune modulation or immune regulating properties there. But during the preservation as well as the transplantation, this endothelial glycocalyx get damaged, and it is due to the oxidative stress and inflammation, and this causes macromolecular leakage as well as kind of like other immune challenges there. So this process kind of like is clearly known that actually like clinical studies have shown that endothelial cell glycocalyx or the vasculature getting damaged, and this is one of the kind of like primary initiating events of any immune rejection happening. So we could, and so we thought actually, since that is happening, and can you kind of recapitulate or kind of regenerate these endothelial cells so that endothelial glycocalyx, so that we'll have a kind of like a better protection of the organs from immune rejection. And if we can do both functional as well as kind of like the steric barrier protections we can generate, we might be able to achieve that. So how we can do that actually? So we thought initially to develop kind of like immune modulating polymers, so that or macromolecules which are attached with ligands, which could kind of like provide the function of the endothelial cell glycocalyx. And if we could do that, so we might be able to kind of like come up with a plan so that it would help to protect the organs from immune attack. So there's a modified glycocalyx. Actually, we anticipated to have kind of like provide immunomodulation similar to the native endothelial cell glycocalyx, and it should have kind of like anti-inflammatory properties, and also kind of like it will provide a barrier for this the macromolecular leakage as well as immune cell binding. So how, to do that, actually, like there is a lot of things to happen, actually. So a lot of tools, actually, we had to develop. One of the first tools, actually, like we had to develop is kind of like a modification which can work in organ preservation or kind of these complex conditions, actually, what we are using. And we don't want to it has to be biocompatible, and also kind of like it could covalently attach molecules on the surface. So for that, actually, like we developed initially, starting with developing a conjugation chemistry which work under organ preservation or organ preservation conditions. So here we are generated a polymer or macromolecules with a peptide probe, and using a conjugating enzymes, we ligated these molecules on the cell surface to generate the functionality on the endothelial cell surface, so that you could kind of like mimic or kind of recapture or re-engineer the GLYCOCALYX. So we looked at a multitude of different types of polymers, and we arrived at the reason why we looked at kind of different polymers. One aspect is kind of like one has to have kind of like provide the steric barrier protection, so that the macromolecules of the cells don't bind to the cell surface. And another option we looked at kind of like can we generate kind of like immune modulating properties, actually. So there we looked at kind of like this sialic acid containing sugars. Actually, the sialic acid is present on the cell surface, including endothelial surface, and which act as kind of like immune regulating molecules. So we collaborated with Dr. Videsh at UBC, and he's a great carbohydrate and enzymologist, and they developed this this molecule actually with the sugar, sialic acid containing sugars. What we did is actually we attached these sugars onto these polymers at different densities and different molecular weights, and attached with a probe which can conjugate, this peptide probe, which can conjugate onto the cell surface. And we call it kind of like GLYCOCALYX mimicking polymers, and it is easily tailorable, as well as it provides kind of like immunomodulatory as well as antioxidant and conjugation properties. So after identifying these molecules, and we looked at how we can conjugate. So where we utilize a enzymatic reaction, where a transglutaminase reaction is an enzyme which can ligate if we have a glutamine containing peptides or glutamine containing glutamine amino acids there, we can form an isopeptide with this transglutaminase. It reacts with the lysine residues on the cell surface to generate kind of an isopeptide. And during that process, you can also generate kind of a side product as kind of like ammonia is generated. So we utilize this conjugation chemistry so that it works at 4 degrees in organ preservation conditions. It works really fast. It works under 30 minutes, and also kind of like works under the complex conditions. So we utilize this chemistry and develop this chemistry to attach the polymers, so the molecules actually be developed on the organ surface for organ engineering. So initially we looked at whether we are attaching these molecules by utilizing endothelial cell monolayers, and we utilized the generation of ammonia, how much ammonia is generated, and that is used for kind of like looking at how much of conjugation is achieved here. As you can see that the molecules with the glutamine, we can conjugate on the cell surface based on the amount of ammonia generated. And also we looked at the cell viability of those, like whether this modification is compatible with the cell surface, or whether it is compatible. So we utilize both kind of conventional biocompatible assays like MDT assays as well as MTS assay. And in addition to that, actually, we also utilize here, actually, I'm showing kind of like the viability data from the conventional assays. It shows there is no kind of like toxicity. And then we also looked at kind of like RNA sequence analysis and other methods to look at in a little deeper into showing that this kind of modification is not doing any kind of like it is well tolerated, as well as actually highly biocompatible. Moving further, we looked at whether this modification is happening at the GLYCOCALYX level or only on the cell surface. So we utilized confocal microscopy look at using labeled polymers to look at whether this surface modification is happening at the cell surface. So it is not taking up the cells, and it is residing on the surface of the cells. Moving further, and we looked at whether this modification is kind of like generating kind of like useful properties, like whether it is preventing or whether it's camouflaging, kind of like if it is preventing the immune recognition, and also kind of like it is protecting from the immune evasion. So here actually we looked at there is a cell surface protein which is ICAM, and which is expressed on the inflammatory endothelial cells, and we looked at whether this polymer attachment on the cell surface, whether it can protect or kind of cover or provide as a barrier for ICAM recognition with an antibody. As you can see that actually like this polymers, when it's attached on the endothelial cell surface, it could kind of like protect the ICAM recognition. And moving further, and we looked at whether this modification can kind of like prevent the PBMC, peripheral blood mononuclear cells, actually whether it can adhere to the cell surface. So you're looking at kind of like a couple of different polymers, and you can see that this modified the modified polymers is protect the cell surface from attaching the PBMC on the cell surface. So which is kind of like interesting at the beginning, and then we looked at further to look at whether this kind of like PBMC adhesion prevention, whether it leads to decreasing the cytotoxicity of the cytotoxicity induced by the PBMCs. Here we utilized the cell surface engineered endothelial monolayers, looking at a few different polymers, and from here you can see that the polymers with sialic acid containing polymers CR3-like3 or CR6-like, you can see that there's a significant reduction in the endothelial cell toxicity can be achieved using this modification. Further, we looked at whether what type of polymers are kind of better, whether the conjugation density of the sialic acid is better, and we looked at different density of sialic acid, and also we looked at kind of like whether this attachment of these polymers on the cell surface is useful, or whether it is kind of a soluble polymer is kind of like effective. But we found that actually cell surface engineered polymers or covalent attached polymers are better in significantly better, in many-fold higher, better active than on soluble polymers. That suggests that actually the presentation of the polymers on the cell surface is an important factor in providing this kind of activity. So further, actually, we looked at whether kind of we could prevent the PBMC mediated cytotoxicity, whether what kind of players are involved. Okay, there are many types of immune cells, and what kind of immune cells we know it is going to be important here. So we depleted different immune cells from the PBMCs, and we depleted NK cells, monocytes, CD8 positive T cells. And what we have seen that actually when we remove the NK cells, actually the beneficial effect is kind of decreased considerably, and that suggests that NK cells are kind of involved in that. So we are continuing the mechanistic understanding of this process, and hopefully actually we'll have better understanding actually in the coming months, or kind of in the ongoing work. Further, we looked at kind of we collaborated with Dr. Levin's group. Actually, they have a CAR T cells actually which is reacted to HLA-A2 antigens, and these antigens are expressed on the endothelial cells, and also kind of these are pretty cytotoxic T cells. And what we have seen that actually using those cytotoxic T cells, and what we have seen that by modifying the cell surface, we could protect the endothelial cell monolayer from the cytotoxic activity of the T cells, which is very interesting in a way that actually like this is a HLA antigens, and which is telling that actually like looking at by providing the right types of surface modification on the cell surface, we are able to protect the monolayer, endothelial cell monolayer. So having seen this kind of like exciting in vitro data, and we moved further and looking at kind of like this activity in a kind of this modification in a really complex environment. Here we collaborated with Dr. Choi's lab in Simon Fraser University, and they have an artery model of transplantation. And what we did is actually like we incubated our cell surface engineering or organ engineering cocktail in the artery, and it's transplanted from biopsy mice to black cyst mice. And we looked at kind of study protocols as a day, after transplantation, the day 2, day 15, and day 42. This artery has been taken out and looked at histologically to look at whether there's any beneficial effect. So the histological slides are shown here, especially if you look at 42 days, you could see that there's a significant reduction in kind of intimal, kind of like the lumen narrowing of the blood vessel is kind of a decreased considerably or significantly compared to the untreated cells. So when we started this work, actually, what we have seen that the coding on the endothelial cell surface is transient. Actually, so what we have, our half-life of the surface of the modification is about 8 hours, but that protection offered by the early protection offered by that coding by about 24 to 36 hours, actually coding is be removed from the blood vessel. But what we have seen that actually like the modification itself at the early stages actually is protecting the transplant from the rejection happening there. And this is kind of like we calculated, we kind of like completed the different the slides, and then you can see that actually the early inflammation actually it's perceived by the medial thickness. And the day 15 actually we look at the rejection score, looking at the how much cell infiltration as well as the thickness of the tissue or the inflammatory tissue. And also we looked at the chronic rejection at looking at the narrowing of the blood vessel during that process. But we can see that actually like this modification is significantly reducing the rejection of the blood vessel, and this modification or this kind of like procedure happen without the need of any kind of like immunosuppressants here. So all these modifications happening without all these studies happen without the immunosuppressants. And we also kind of like seen that actually like on day 2 inflammation. So we could see that the major cytokines as well as anti-inflammatory cytokines and chemokines are anti-inflammatory chemokines are expressed more, and pro-inflammatory cytokines are kind of like decreased significantly in the day 2. And this is kind of like and there is kind of a good correlation between the pro-inflammatory cytokines and then the antibody-mediated rejection, which is kind of like reported in the literature previously. And what we have seen that actually like in the day 42, we could see that the donor-derived antibodies are kind of significantly reduced in the recipe in mice. And you can see that the treated or kind of cell surface engineered the grafts do produce less amount of kind of like antibodies against the donor grafts, and which is kind of like interesting in a way that actually we did this modification, and this modification is very transient, but still that effect is kind of like persistent for a long period of time. And we further looked at kind of like, moved further to looking at kind of like a renal transplantation. Here we did 2 studies. One study actually looking at kind of like a SYNGENIC transplantation model, where actually we want to kind of like eliminate the issues with kind of like the immune system, so that actually we utilize a SYNGENIC model here to look at whether kind of like the protection of the transplant is how the protection of the transplant is good with this kind of modification. And we collected the kidney and is perfused with the solution, and it's for this study's been done with static storage for 4 hours to generate kind of like sufficient injury, and then it's transplanted. And you can see that this mice with this modification actually can be protected really well, and that is shown by kind of like this showing kind of like cell infiltration to the tissues, kind of like those organs kind of like decreased, and also kind of like pathological score is also telling that actually like the organ is functioning better. And this is done in collaborations with Dr. Du and Dr. Sang, and Dr. Sang has got an excellent model for renal mice transplantation. We further looked at kind of like allografts. Here, the same procedure actually is done at 1 hour modifications, and then transplanted from a biopsy to B6. You could see that actually we can protect the organs, and the kidneys functioning better in terms of like blood urea level, and also kind of like cell infiltration can be kind of like significantly decreased by this modification. And this is kind of exciting. And then we looked at kind of like whether this immune suppression is whatever the immune suppression what we are seeing, whether it is local or kind of like whether it has got the global effect is there. So here we looked at kind of like a double transplantation model, and he started from artery transplantation to the black cyst mice, and then we waited for 28 days, and then we transplanted that mice with the skin transplantation from another mice, and looking at kind of the survival of the skin transplant. And our hypothesis was that if there is a global immunosuppression, and this skin transplant will be surviving longer, and if it is not, actually like this, it will have kind of like similar it would reject immediately. And the skin transplant is not kind of modified with CSE, and but only the modifications happen to the artery. So what we have seen that the untreated and the treated do have kind of similar kind of like survival, which is suggests that actually this modification, what we are the organ engineering approach actually we are developed is acting as locally, and that local effect is kind of like providing this beneficial effect on the transplantation. So where we go from here actually, so we are looking at different organs, and we are also looking at kind of like whether cells can be modified, and islets actually we are looking at, and then wound healing as well as other body function properties, and we are looking into that. We are also kind of like developing this further, looking at new immunomodulatory molecules, and we are also looking at kind of, instead of attaching the polymers, and we are kind of like growing these polymers from the cell first. It has got some beneficial effect, which since it is not published, actually I don't want to go into details there. And we have seen kind of like beneficial effect of kind of like growing polymers from the cell surface too. So in summary, and what I have shown you, kind of like we developed a novel organ engineering approach using cell surface engineering, so that actually we are able to kind of like produce using kind of like immunomodulating polymers. We are able to kind of like provide, regenerate kind of like the damaged GLYCOCALYX, so that actually like the transplant can survive better, and we have a better protection of the transplant is achieved. And our hope is that actually, like, since our work, although it is kind of in a very early stages, it shows kind of like we haven't used kind of immunosuppressants here. And so we might be able to, using this kind of approaches, one day actually, like, we might be able to kind of like maybe eliminate, or kind of in the other case, actually, we can minimize the use of immunosuppressants possibly. And the cell surface engineering approach we developed, that is kind of the crucial for that, working at organ preservation conditions, and this modification can be worked during the kind of a preservation or preservation, preservation's time, so that actually the clinical translation is kind of little more easier. And before concluding, I would like to thank my group, current group, and also collaborators which are essential for this project, including Dr. Vides, Dr. Choi, Du, and Chris Nguyen, and Dr. Sang for kind of like many of this work, and different funding agencies, and which made this one funding working, this work possible. And before concluding, I would like to thank you everyone for listening, and I would like to take questions you may have. Thank you very much.
Well, thank you, Dr. Kizhakkedathu, for a wonderful talk and stimulating and real innovations. I welcome all participants to either raise their hand or add in their questions into the chat. And as they start to trickle in, I could start with a few questions. Well, we all know that temperature does affect enzymatic activities. I was wondering if you could comment on the effect of temperature on your cell surface modification, apart from, say, normothermic ex vivo machine perfusion for organ preservation. Most are still utilize cold storage. So did you see a difference with temperature? And is it possible to use this in ex vivo machine perfusion?
So that's an excellent question, Dr. Pepper, actually. So this, when we started, actually, like we wanted to develop we looked at kind of like many different surface modification strategies. So chemical modification as well as enzymatic engineering. So chemical modifications usually very sluggish at low temperature. Okay? So that, and also kind of the reagents what one could utilize is kind of like not great actually. So that's the reason actually we moved to kind of like enzymatic engineering approach. So in the enzymatic engineering approach, one of the first goal was kind of like, anything works at low temperature, kind of like 4 degrees actually, like for us, our first goal, okay. And what we have seen that actually like, our modifications were, great at, excellent at, 4 degrees. And also kind of like it can work from all the way to 4 degrees to 37 degrees or more, okay. So without much change in the activity, okay. There is some slight change in the activity of the enzyme, at, it increases activity if you increase the temperature. So even if you wanted to use EVLP protocols or other things, this is not going to be a problem at all. And this kind of like modifications work beautifully at very different temperatures.
Wonderful. I see a question from Murray Wilson. I apologize, this is a naive question. However, how do you move this from theoretical science to benchtop testing to clinical studies, which can into medical journals? Murray, feel free to unmute yourself too to follow up if you like.
Okay, thank you. Thank you for that. This is an excellent question. So we showed that actually like this is works in in mice, mice kidneys. Okay, I could say for a transplantation area actually this is kind of very early stages. So our plan is kind of like, is to going from pig studies, that is kind of our next stage, and then a non-human primate studies actually like, after that, okay. So that's what actually we are looking for. And in the clinical stage, actually, what we are currently actually talking to, FDA and other people, our kind of like contacts there. And what the suggestion is kind of like these modifications could be potentially, can be gone as kind of a biomedical a device, okay, and similar to kind of like organ preservation and EVLP, solutions, and LVAB protocol actually all approved as kind of like a medical device. So most likely actually like this will go as a kind of medical device since it's a transient modification, but I cannot guarantee that actually, but we have to go and talk to them, okay, with more data. But this is our clinical path look like. We are going for the pig studies, and we'll be doing kind of like both allografts as well as kind of syn graft. The reason being is actually like it has got kind of like perhaps actually it gives kind of better protection, and also kind of we'll be doing in combination with immunosuppressants. That is another. So going from there pigs to non-human primates, and then in a clinical studies. That is a plan.
Wonderful. There's 2 questions from Andrew Masoud. Thank you for presenting such an inspirational research work with this stupendous display of such techniques. The first question is, are there any updates on utilizing the cell surface engineering modalities to manipulate the cell biology during cellular senescence? For example, the pro-inflammatory senescence associated secretory phenotype.
I think possibly actually we are just beginning to explore this. Okay, so it's kind of like, you know, and we might be able to kind of like you know, present different types of molecules on the cell surface with this kind of techniques. The chemistry developed actually is adaptable for kind of diverse applications. So what I showed actually, like one type of molecules can be presented, it shows activity. But similar to that, actually, you might be able to present kind of a different molecules of interest, and that could possibly do that. But whether you know, where the modification is going to happen is kind of maybe an important question relevant to this question. So in our case, actually, we are utilizing you know, GLYCOCALYX or the sugars modification. So but if you have seen that actually that sugars will be removed during the membrane turnover process or endothelial glycocalyx shedding. But if you can modify the proteins during using this transient modification, so those kind of like the you know, applications, actually this kind of like surface engineering could be beneficial there, and people could utilize both you know, kind of genetic engineering in combination with this one. You know, that is another possibility. We could do that. I hope you answer your questions.
Great. And the second follow-up to that is, what is your perspective to speculate on this approach in cancer cell or vascular cells in degenerative diseases context?
Okay, so in terms of GLYCOCALYX editing, I could say that actually there is a, you know, there's a lot of interest actually going on in that. Okay, like, you know, cancer cells do express you know, more sialic acid on the surface in a way of kind of like preventing the damage, or kind of prevent the elimination from the immune system. So there is you know, people are looking at kind of knocking down the transferase expression, so that actually you might be able to generate better medicine. There's few companies working on that too, and there is a lot of interest there. Okay, there, and regenerative medicine case, and there is a lot of papers recent papers are coming on GLYCOCALYX editing and how the GLYCOCALYX editing can possibly kind of like enhance the survival of the tissues, especially the muscles and other tissues. Actually, people have shown, and we are also looking at kind of like you know, a skin transplantation, for example, you know, in our purposes, and also and a few other applications. Actually, we are also looking into that.
Great. There's another question from Murray. I don't know if he's going to open up his pockets with this question, but he says, Dr. J, how much funding do you need in the next 3 to 5 years, and where will it come from? I guess.
Okay, Murray, thank you very much. You know, we would welcome a lot of funding from you. So I was looking at actually how much a pig studies would cost us. Okay, minimum $1 million for a pig studies for us. Okay, so that is what kind of like to show the proof of concept, and then beyond that, actually, like, you know, you could say like, you know, 10 to $20 million actually could give us kind of like you know, going until the clinical studies. Actually, that is what my thinking. including the non-human primate study. So I'm looking for that kind of funding, and let us know actually, like, have some contacts actually I could talk to.
Dr. J, I'm on the board of TRFBC, as well as a co-founder of the Alberta Organization Group. You should. Does TRFBC know what you're doing?
I don't know. Okay, I do talk to CDTRP. Okay, and I do talk to them, and they do support our work, and they gave us
there's a
an
joint
excellent
there's a joint venture fund with TRFBC, but I don't know. I haven't heard your work before, and I think it's absolutely fascinating. So, make sure that TRFBC knows at VGH-UBC, and you can contact me whenever. I think this work is absolutely incredible. Thank you very much for the presentation.
Cool. Thank you very much, Marie. Actually, this is exciting. So I'll call them and let them know. Okay, thank you.
Thank you, Marie, for that connection. And speaking of pigs, have you thought in recent news with xenotransplantation, with the heart donator and then the descent model with kidneys, have you thought about this in xenotransplantation, reducing hyperacute rejection?
I think that's it's an interesting connection there, actually. So this, in my mind, actually, like, in our collaborators, actually, we talked, and this modification, irrespective of actually what kind of genetic modifications we do during the transplantation, the GLYCOCALYX or the endothelial cells can damage. So irrespective of what mode of transplantation, whether it's a xenotransplantation, whether it's a vascular transplantation, whether you are tissue engineering and a vascular, a blood vessel, whatever actually like the conditions are, actually this technology could be applicable there. It has got a broad applications. I'm sure I'm kind of like this. If this works in pigs and non-human primates, this would have kind of a lot of implications in all the forms of transplantations.
Right, especially tethered with some genetic engineering of potentially the pigs, CRISPR pigs alone, that may not be enough, especially looking at hyperacute rejection. So that's great. And just one other follow-up question. In your skin graft model, when you had the C3H reject, did you see any effect on your vascularized graft? Did that precipitate rejection of your transplanted graft?
Yeah, that's a good question. So we looked at the histology of the graph. Okay, so we've seen a kind of almost identical, behavior as kind of like similar to our original graphs. So the original protection. So that original protection is kind of like really, keeping it there.
Great, it's pretty stringent. There's another question from Andrew. Is it feasible to collaborate with ex vivo perfusion teams of the transplanted organ before transplantation to apply a CSE modification in effort to preserve and enhance organ survival? And thank you for answering all the previous questions.
Okay, of course, actually, we are starting to collaborate, okay, and on that. And we have a collaborator on lung transplantation with Dr. Seipel from University of Toronto, and he is developing a protocol on that. And we have a recent paper came out from their group in our collaboration with Laurie West and Dr. Vitez and all our group in Science Translation Medicine, recent paper on ABO conversion using EVLP protocol. And we are beginning to start that collaborations on lung transplantation. But and there is nobody in Canada actually doing kind of like EVLP for kidney transplantation. If I know a few people are beginning to do, and if you have anybody contacts, I would be happy to talk. And then there will be, I have a lot of interest in doing that kind of studies.
It may be worth reaching out here to the U of A with Darren Freed and Dr. Jayachandran for heart and lung ex vivo perfusion.
Okay, great. Thank you.
Great. Well, I don't see any more questions, so I'd just like to thank you for sharing with us your innovative research, and the multi-applications that it can be applied to, and stimulating some wonderful discussion. I know from an islet perspective, my mind's been racing quite a bit. So hopefully maybe we can contact each other in the near future. I just have one closing announcement in regards to next week's ATI seminar on April 6th. We're very pleased to have Dr. Phil Halloran presenting molecular rejection and injury states in organ transplantation populations. So there's a link in the chat to that Zoom to register for next week's seminar. So give about 5 minutes back to everybody's day today, and thank you once again for joining us.
Thank you very much.